Okay. I have. Does anybody have the key up?
I have the key up.
Yeah.
So number 1 says transcription in E. coli is subdividing to 3 stages: initiation, regulation, and determination. What DNA sequences are involved in initiation in E. coli? This is going to be a lot harder than. I feel like anyway, this is going to be a lot harder than anything else. But I said the 17 base pair region, the -35, and -10 sequences. What did you guys say?
Wait, for part A or? Yay, Eileen!
Hi.
Hi.
Just gonna be like really bitchy all day. Don't even worry.
Alright. No worries.
So, what about number 1?
So, what did y'all get for number 1? A.
1A. Apparently, I got that really wrong, according to Sister Mayo. So, I probably shouldn't talk.
What did she say the answer
Wait,
was?
no. Oh, wait, no, JK, that was 2A. That I got wrong, not 1A. 1A, I just wrote regulatory DNA sequence, promoter, and terminator sequence.
So, regulatory, promoter, and terminator.
Meant 2A. Yes.
Were the 3. Let me.
Yeah.
I'm looking at the key and I'm realizing that the answers are really long, but I think like I just answered like just what it asked for, not like where it came from, if you know what I mean?
Yeah.
Yeah, mine's not even a sentence.
Yeah.
Yeah.
So, regulatory sequence, promoter sequence, and what was the third one?
Terminator sequence.
Terminator. Well, dang it. Okay.
Yeah, sorry, I meant 2A. I got wrong, not 1A. I looked at the wrong one. My bad.
That's all good.
But yeah, so that's what it said. And then I gave a giant lengthy paragraph about, I think about what it is, ... I didn't read the whole thing because I didn't feel like it. Like, in the...
I would think that the terminator protein would be not involved in initiation, but termination. Wrong? I mean, obviously I'm wrong, but...
I don't know.
Okay. 1B.
Okay, cool. What proteins are involved in the initiation of E. coli?
RNA polymerase, holoenzyme, and sigma factor.
Yep.
Yep.
Sweet.
At least I got that one right.
I know, right? I'm with you. Okay.
Cool.
What proteins are involved in elongation in E. coli? Just RNA polymerase?
Yeah, the answer is just RNA polymerase, but I wrote like 2 more things on top of that, so I got that wrong.
Would it make a difference if you like just put RNA polymerase instead of RNA polymerase core enzyme? Because it says core enzyme, so I don't know if.
Yeah, this is the first science class I've taken that it calls it a core enzyme. All the other ones, they just call it an RNA polymerase.
So it's the same exact thing.
Well,
Yeah, I think so.
Like even in 265, or no, is it 181? It's just RNA polymerase.
Okay.
What DNA sequences are involved in the termination in E. coli?
The terminator sequence.
That's what I thought. But like isn't it different?
Is it different?
Depending on like I know the RUT sequence is involved in row-dependent termination. But I don't know a sequence that's involved in row independent. What does the key say?
It just says terminated sequence and then talks about stem loop structure. And I mean, it does mention both row dependent and row independent. So I guess if we wrote row dependent independent termination, then I guess it's correct. But overall, as a thing, it just says terminated sequence. So I think either ors are okay.
All right. And
Wait, for that one, it's like there's a terminator sequence involved in both, but they just terminate differently. That's why.
Oh, okay.
Mm-hmm.
Yeah. So it's the same like sequence. It's just like how they do it.
so it's just the answer is the termination sequences there.
Yeah.
Okay. E, what proteins are involved in termination in E. coli? Or like the row protein, right?
Yeah.
Mm-hmm.
Or
It also says. Sorry.
no, go for it.
It also says NUSA or NUSA or however you say it.
Is RNA polymerase involved?
No.
No,
No.
at least it doesn't say it.
Okay.
It like mentions it, but it doesn't.
Yeah.
It's not like a main
Answer.
one. Yeah.
Okay, who wants to do two?
Okay. What is the function of a sigma factor? When the main question asks about the family of proteins of sigma factors. So.
It binds with a core enzyme to form hollow enzyme and binds to the -10, -35 sequence in the promoter.
Yep. Yeah, you got it. You mentioned all the things that were needed. I literally just wrote binding site, so she was like, no. Because I forgot all that key stuff, so that was my bad. Okay.
B confuses. I'm interested to see what the answer for B is because, yeah, I don't get it.
Okay. Should I read it, or does anyone else want to read it?
I guess I can read it.
Yay. Okay.
This is straight from the key. Housekeeping refers to all cellular functions in E. coli that are required for life. Therefore, the housekeeping sigma factor functions to guide the RNA polymerase core enzyme to the promoters of all genes essential for life in E. coli. This ensures that these essential genes are continually transcribed.
So it guides the polymerase to what?
It
It says to the
to
promoters.
the RNA. Yeah.
Yeah.
Guides the RNA to the promoters, the RNA polymerase to the promoters of all genes.
Basically, I looked this up online because I didn't know what the answer was either. And I found stuff that said that it just keeps the gene pathways open. What's on my camera? Something. Sorry, you guys. It's really blurry because there's something. My camera's all dirty. But yeah, it just keeps gene pathways open so that it can keep functioning. So it can keep transcribing. The key is just more specific with how it does it, but that's all it does.
Okay.
Sounds good.
Do we want to move on to C?
Yeah.
Yeah.
Cool. Other sigma factors in E. coli included general starvation, nitrogen starvation, and excessive heat. Sigma factor of what benefit is having multiple sigma factors? There's a giant paragraph, but I'm lazy. But overall, it's just to respond to stressful environments and just to adapt easily, so that way it can change whenever the heck it wants.
So
Yeah.
it's more resistant.
Yeah.
E. coli is cool like that. Yeah, jazz hands. Okay.
And D says sigma factor is a good example of a transcription factor. Provide 5 other examples of transcription factors.
Yeah, I just took that one straight off of slide 14.
Oh, okay. I need to look at slide 14. I'll put a note there.
I don't know if it's right. I haven't checked, but that's where I got it from.
Yeah,
All the
yeah, it's close
TFs.
by. It's the one on slide
Yeah.
14. So you're good.
So just all the TFI IDs then?
Mhm.
Yeah,
It's also in
B,
the notes.
E, and H.
Yeah, that's what I put. I only use one of the TFI IDs, but I put alpha, beta, beta prime, and W.
Yeah.
I can't remember what that is. Do those count too?
Did she list more than 5 just so that we could know? Let me check actually. I should just check.
No, all she wrote was the TFI
Yeah.
ones.
Oh yeah.
That's all she
Well,
wrote.
that's just
But there can be more, yeah?
I think so. At least I found more.
Oh,
because
I found
Get the right, that is.
and eukaryotic, and TFI IDs are just eukaryotic, right?
Yeah, TFs are eukaryotic. I didn't hear the rest of your question, but I'm gonna say yes.
Okay. You guys want to go 3?
Yay. Okay. Number 3, we learned that E. coli terminates transcription using either the P dependent, dependent, or P independent. First one of A is, how are these two mechanisms different?
The use of P protein.
Yep.
Wait, different? Yeah, different.
Different, yeah. The 3B is how are they similar, but 3A is how are they different. So yeah, it's how they use the P protein, as you said. So you're good.
Guys, it's a row.
It's
Just
a what?
saying. It's a row. It's not a P.
It's
It's a what?
I guess
It's the Greek letter rho.
Oh, really?
Yeah, that's what row independent and row dependent is. That little P is a row.
oh the P, not the
Oh.
Yeah.
Now I feel stupid.
I'm just correcting you.
Like, the whole time I've
Just-
been reading it that way. Like, it's stuck in my head now.
yeah, it's a lowercase letter.
That makes a lot more sense. Okay.
Yeah, for why there's like row independent and row dependent.
Mm.
But yeah.
Thank you, Eileen.
Sorry, I'm in
Like,
such-
now it's like stuck in
such
my
a good
head.
mood. What?
Because now it's like stuck in my head because that's how I've been reading it.
Oh, that was a little P. No, there's like in physics it represents density. So, or I guess in chemistry too.
Yeah.
So like a row usually is a density. It's a letter that represents density. But I guess there's the genetics representation for row too. That's cool. But yeah,
Yeah, I guess this shows how much we know our Greek letters.
Definitely.
Yeah.
I don't know. This is just one I learned when I was like 19, so I just have never forgotten it. But,
I don't know, but recently I've been seeing a lot of sigmas.
yeah,
With
yeah.
class and chemistry, so.
Seriously.
Mhm.
Ew.
So then 3B,
Okay.
Yeah.
or yeah.
Yeah, now that we know it's a row,
Yeah.
dependent, independent. I feel so
Like,
stupid.
for A, independent, it doesn't require the row protein. It has uracil-rich regions, and it has the it uses the Nus A protein. And row dependent requires a row protein that has a RUT sequence, right?
I believe so, yes.
So like the way that I understood this is like row dependent because the base nucleotides are C and G. Those form like a stronger bond, so you need that go in and break that up. But in a row independent situation, it's the ending, like the terminating sequence is adenine and thymine, and those things form, or adenine and uracil, and those form like really weak bonds, so it doesn't need a protein to go break it up. It just like falls apart at the end.
Well, that makes more sense.
Yeah.
You got this in the bag.
Yeah, I'm in trouble.
Oh my gosh, you guys, I feel like I haven't done anything. I feel not prepared for this test. Ugh.
More prepared than I am.
Ugh.
I'm more than
After this, that's all I'm going to be doing is studying for the test and hopefully taking the first part today.
Yeah. Ugh.
Hopefully.
Yeah.
Hopefully.
I have a GIS project due tomorrow, so I need to get that done, and I'm just like freaking out because I have all this stuff to do.
You can do
Yeah.
it.
The end of the semester, guys. Everything is due.
Yeah, just failed the chemistry test this morning miserably, and so that was fun.
That happens. Even the smartest people like fail chem. Is it 106?
It's 105.
That's even worse.
Yeah. As long as I keep my B minus, I'll be happy.
Who's your teacher?
DeBall,
Oh, okay. I never had him for anything.
he's very intense teacher.
Hmm. Oh man.
Should we go to B?
Yeah.
Sure.
Oh yeah, sure.
Well, I think we covered it actually. We covered how they're different and how they're similar.
Okay.
Well, I mean they're similar because they both have the stem loop structure.
Yeah.
And there's one more.
No, that's
I
actually
only
it.
mentioned
That's all the
the
key
stem loop.
says.
I know, but I put one more thing that I found that they were similar in. Hold on, let me find it.
That's pretty
Oh, I just said that they're like similar but different because they like both use their nucleotide base pairs for their like dissolution, but they use them in different ways. So yeah, kind of like what I said. Like one uses the GC base pairs and one uses the AU base pairs. So it's like they both are using those base pairs to dissociate, but they have to have different types of proteins and like different ways of doing it.
makes sense.
Okay.
All right, sweet.
Okay. I guess someone could read.
See.
My battery's about to die, so if you see me disappear, that's why.
Okay. So C says, suppose you're studying row-dependent transcriptional termination in the lab. Propose 2 ways that you could interfere with the row-dependent transcriptional termination process. In terms of specific changes you could make to the DNA or messenger RNA that would inhibit transcriptional termination.
I hated these 2 questions.
I don't know if I got them right, but I thought it was like applicational, you know?
Yeah, I just couldn't figure out like what to change and what's going to come
Or
out
what
of
you
it. I guess I
could
tried
do,
as
that sort of thing.
simple as I possibly could.
So like we could delete the rut site
Mm-hmm.
in RNA.
Or
I
like
think that's what I
change it,
mentioned.
like change the rut sequence. That's like what the key says, like to create mutations in the rut sequence, but you could just change it.
Yeah.
Mhm.
Could I see the thing is I don't know what we're like allowed to do. So like we could take out the stem loop if we're allowed to do that, and that wouldn't I can't talk. Then it couldn't slow down in order for the row protein to catch up.
That's like what the second part of the key says. It says create mutations in the terminator DNA sequence that prevent the formation of a stem loop. So you just like make it so the stem loop can't even be formed, and then that like it'll prevent both kinds from happening. So like it says changing one of these GC rich regions to AU rich regions will prevent stem loop formation. So
So
like
that's right?
what?
So it's right?
Yeah.
Okay.
Yeah, so find a way to take out the stem loop and then find a way to take out the rut or like change the rut.
Cool. Alright. And D says, suppose you're studying row independent transcriptional termination in the lab. Propose 2 ways that you could interfere the row-independent transcriptional termination process.
Like the same thing.
Specific changes you can make the DNA or mRNA that would inhibit. So it's like the other one.
Yeah. So again, it says change the stem loop thing, like either change the formation of the stem or prevent the formation of the stem loop.
Mm-hmm.
So same as the one above. And then the second one you could do is to change the terminator sequence from AU to GC, and then it won't be able to dissociate.
Okay. Cool.
Yep. I don't know how would you do that stuff though without like changing, like, because every gene codes for, or every like, like gene set of base pairs codes for a protein.
Mm-hmm.
So like I don't know how you would do that and change like the GC to AU without changing the proteins that are expressed. You know what I'm saying?
Yeah.
Like, I don't know. I feel like you'd end up like changing a lot more than just like you think you're just changing a couple base pairs, but you'd probably end up changing the protein and then like the overall phenotype of whatever that DNA codes for.
That's what I would think too.
Yeah, it's weird. It's probably not a good idea.
Okay.
Okay, number 4 says eukaryotic genes can be transcribed at the basal level or can be transcribed at levels above or below the basal level. Transcription above or below the basal level is called regulated transcription. So part A says what DNA sequences are required for basal transcription in eukaryotes?
The TATA.
Yeah.
In the +1 site.
Yeah. See, and then yeah, so both those are right. And then it says what proteins are required for basal transcription in eukaryotes.
It's like the same ones we
So
said
it's like
before.
those 5 transcription factors from before, the TFII ones, and then RNA polymerase 2.
Okay.
And then C says what DNA sequences are involved in regulated transcription in eukaryotes.
The CAAT box.
Yeah.
Yeah.
And then GC and how do you say that? Octamer boxes.
Is that all that was on the key?
It just says promoter elements, and underneath that are like the CAAT, the GC, and Octamer.
Okay.
So promoter elements, core promoters, and regulatory DNA sequences, so enhancers and silencers.
Is that D?
That's C.
C.
Okay. Oops.
So yeah, there's like a bunch. And then D is what proteins are involved in regulated transcription in eukaryotes. And again, it's RNA polymerase 2, the same 5 general transcription factors. And then regulatory transcription factors, so activators and repressors, mediators. Is that it? Yeah.
Okay. And 5, this question concerns the 3 major RNA modifications discussed in this lesson. A, suppose a oh wait, did I just... Yeah, okay. A, suppose a pre-mRNA is produced by transcription. Which RNA modification occurred first, which RNA modification occurs second, and which is the final RNA modification. So I said first is splicing, second 5' capping, and third is polyadenylation.
It says in the key that the first one is 5' end capping,
Okay.
and second is 3' end polyadenylation,
What's the
and
third?
then the third is intron splicing.
Oh, oops.
Mhm.
I guess that would make sense because at the end you cut it out.
Yeah.
That would make sense.
Yeah, because there's yeah. Yeah, never mind that. Yeah.
That makes more sense. So the ribozymes play an important role in RNA modification. What is a ribozyme, and which RNA modification process involves ribozymes?
Intron splicing for the modification.
That's what I got too.
Yeah. And it says it's a catalytic RNA molecule.
Sweet.
Cool.
Is that all that it's involved in? Is intron splicing?
Yeah.
Yeah, that's it.
Sweet. See, what is the function of each of the following molecules in RNA modification, the S and R and Ps?
Yeah. Let's see.
Removing introns.
Yeah.
Yeah, it's one of those spliceosomes complex subunits of it.
Is that all it's responsible for, is removing introns?
Yeah, that's all it says.
Okay, sweet. Guanosine nucleoside.
The key says that it serves as a cofactor for group 1 intron splicing.
Okay. For what kind of splicing?
Group 1 intron.
Okay.
Bless you.
Maybe I should write that. I should just check the key is what I should do. CPSF.
It says it's involved in 3' polyadenylation. It recognizes the polyadenylation signal sequence in the pre-mRNA and cleaves the pre-mRNA 1035 nucleotides downstream of the polyadenylation signal sequence.
And poly A polymerase. It attaches adenine residues to the 3' end of the transcript. Is that the answer?
It attaches what?
It attaches multiple adenine residues to the 3' end of the transcript.
Yeah. It makes the poly A tail.
Okay, I should write that down.
But yeah, that's right. And it says about 250 bases of adenine.
Okay. Guanylyl transferase.
So this one says that it's involved in 5' end capping and that it cleaves GTP to produce GMP and PP. Is that an I? A little PPI.
Pyrophosphate.
Yeah. It says it's then the GMP is covalently linked to by guanylyl transferase to the 5' end of the pre-mRNA transcript.
Okay. Awesome.
Looks like I'm going to have to use all these keys to study.
Yeah.
I always use these to study.
What does y'all's picture look like?
I haven't done this. I thought this was another one we were doing together, but maybe
Yeah.
I'm just lazy.
Yeah, like Because that's what I understood. It's like from now on, basically, is number 5 we're supposed to do together.
Sorry.
That's
Is-
okay though, because I'd rather not sit here and all draw it together because it's
Yeah.
really boring.
I just drew what was on the slide with like, I don't know if this is right, but
What slide did you use? Oh wait, I need to open mine.
I don't remember what, I think it was like the last slide number, but it had something
Oh,
like,
does it have an R?
Identifying
Oh,
introns.
that loopy-loop thing.
yeah, I think it's that because that's the 2 introns. I would think, I was kind of confused, but
Okay.
anybody
Yeah, I'm
have
not gonna lie,
anything they'd like to discuss?
I don't think so.
Okay.
Yeah, my hardest thing
Good.
was that slide right there because I didn't know what I was talking about and I purposely gave up. But now that makes sense. Now that you showed that.
Oh, I'm glad because it doesn't make sense to me. But I'm glad it makes sense to you. But
Like I got
okay.
like a semi idea of what we're talking about because I just looked at the paragraph of the question. I was like, nope, that's a chapter in our lives. We're done.
Does she have a picture of it in the key?
Nope.
Oh.
She just said I will draw it.
Okay, well, hopefully we get that soon. Okay,
Yeah.
well, I will post the link if you guys are done.
Okay. Thanks, girls.
Yeah, sorry, like, this is gonna be a hard one.
I gave up like after 5B. 1 because I was tired, and 2 because it was just hurting.
It's just hurting.
Okay,
I'm gonna watch all the
well, I'll
videos
post
again.
them. Oh, what was that?
I said I'm just gonna go watch all the videos again.
That's a good idea. I haven't watched the videos yet.
Have any of you started your exam?
Mm-mm.
Yeah.
I'm gonna do it tonight.
I'll probably do mine tomorrow.
Same here.
Just watch all the videos today and then do it tomorrow.
Yeah, exactly.
I kind of want to read through all the packets too.
Mm-hmm.
That really helped me with the last quiz, like reading out loud. It probably drove my roommates crazy, but like reading out loud the packet before the notes before I went and took the quiz, I got like a 90, and that's the highest I've gotten.
Nice.
Wow.
And-
I can't read out loud. That does not work for me.
Okay.
Well, thanks.
Yeah, y'all have a good day.
Just do.
Bye.
Bye.
Bye.